Hi guys, Miss Francis here today to discuss the relationship between cancer and mitosis. Well, cancer is uncontrolled cell growth and cell division, which in essence is mitosis. So cancer cells can survive indefinitely if they have a continual supply of nutrients, and that makes them different from normal cells because normal cells will not be immortal. The first immortal, so to speak, cells were taken from a tumor removed from Henrietta Lacks. So Henrietta Lacks was a woman who went to Johns Hopkins University to get treatment for ovarian cancer. And the doctor at Johns Hopkins took her a biopsy of her tissue and then sent it to his colleague, Dr. Gay. And then from there, Dr. Gay had taken samples from all patients that were getting treatment at Johns Hopkins. And what he realized was that these HeLa, as they're called, HE for Henrietta and La for Lax, the HeLa cells did not die like all of the other tissue cells did. What they did was actually double in like every 20 to 24 hours. These HeLa cells played a significant role in developing the polio vaccine, in exploring what happens to cells in zero gravity, as well as used in developing cloning techniques, gene mapping, and in vitro fertilization. If we were to compare a normal human female karyogram with a HeLa karyogram, you'll notice that there are additional chromosomes located at certain positions. So that's the difference between a HeLa karyogram and a regular female karyogram. A mutagen is a physical or chemical agent that changes genetic material. It changes the DNA. In other words, it mutates the DNA. Some mutagen examples would be radiation. That's why when you get an X-ray, you wear protective material. Formaldehyde is a toxin that's considered a mutagen. Oncogenes are cancer-causing genes. Now, everyone has oncogenes. It's just a matter of whether or not these oncogenes get mutated or not. And tumor suppressor genes stop tumor development, where tumor is considered a mass of abnormal cells. Now, you can have two different types of tumors. You can have a benign tumor and a malignant tumor. Benign tumors are usually harmless. They can be removed by surgery, whereas malignant tumors have metastasized, meaning that the deformed or defective cells have traveled to other parts of the body and have impaired the functions of one or more organs. The treatments for cancer that has metastasized includes radiation and chemotherapy. So these treatments target actively dividing cells. The issue with these therapies is that there are severe side effects. In current time, in present day, we are working on gene therapies as well as immunotherapies to help people who have cancer. Now, where does this cancer, this uncontrolled mitosis even come from? It can come from faulty signaling pathways, from mutations in the human genome, for example, the P53 site, where P53 is an example of one of those tumor suppressor genes. If there's a mutation in a tumor suppressor gene, it can no longer suppress that tumor. This is a nice little review of cancer and stem cells. Let's start off discussing cancer. So cancer cells may divide indefinitely if they have a continuous supply of nutrients. For example, HeLa cells were the first identified immortal, so to speak, cells, where cancer cells don't exhibit contact inhibition. So most cells normally, once they bump into, so to speak, their neighbor, they're going to stop growing and dividing. But cancer cells don't recognize their neighbor. They don't exhibit contact inhibition. In other words, when they're in contact with another cell, the growth is inhibited. Therefore, the growth continues and the cell division continues. Now, there are different types of cancer genes. There are oncogenes, tumor suppressor genes, and then DNA repair genes. Oncogenes normally stimulate growth. Everyone has these oncogenes, but what happens when an oncogene becomes mutated is that it makes cells continue growing. They don't stop growing or dividing. If we were to evaluate this as a car, an accelerator stuck to the floor would be an example, or would be what a mutated oncogene would be equivalent to. Tumor suppressor genes normally inhibit growth. What happens when a tumor suppressor gene becomes mutated? Well, growth is not inhibited, therefore tumors form. The car analogy to remember a tumor suppressor gene would be like a dysfunctional brake, where if I were to press the brake, the tumor would stop growing, but the brake isn't properly functioning. P53 is an example of a tumor suppressor gene, and it acts as an on-off switch for cell division. So basically what it does is it halts the cell cycle if DNA defects are detected, which enables DNA to be repaired, and then the cycle restarts itself with this repaired DNA. But if P53 mutates, then cell cycle proceeds without the DNA getting repaired. So what do these DNA repair genes do? Well, they limit mutations. But if those genes, those DNA repair genes, are mutated, DNA doesn't get repaired, which leads to those mutations, where if I have too many mutations, it can lead to cancer. So the analogy, if we continue with our car situation, would be like a mechanic. But if my DNA repair genes are mutated, that's like an inept mechanic, an ineffective mechanic. Now to review stem cells, if we were to talk about two characteristics of stem cells that distinguish them from body cells or somatic cells, stem cells are undifferentiated or unspecialized, whereas body cells are specialized. Furthermore, stem cells retain the capacity to divide, whereas body cells may not. If you were to take a look at a therapeutic use of stem cells, for example, stem cells obtained from bone marrow are used to treat leukemia, where the patient's bone marrow cells are replaced, and then ultimately they're able to produce healthy blood cells.